Really super excited to have Reto Fiolka talk at our Janelia and EMBL bioimaging seminar today. He's a Janelia alum in computational fluid dynamics, where he got his master's at ETH Zurich, and then he completed his PhD also at the ETH Zurich in the group of Andreas Stemmer in the Institute of Nanotechnology, doing advanced imaging there. And then, as I said, he's a Janelia alum. He worked with the late Dr. Mats Gustafsson from 2009 to 2012, doing 3D structured illumination microscopy, and then did adaptive optics afterwards. In 2016, he won the prestigious Cancer Prevention Research Institute of Texas a tenure-track recruitment award, and he started his tenure at UT Southwestern Medical. And his lab is interested in imaging in physiologically relevant 3D environments, basically across all scales from subcellular imaging to the mesoscopic scale. And he's really working on improved spatio-temporal resolution, as he will tell us today, in fluorescence microscopy, light sheet fluorescence microscopy. And with that, I'm going to pass it to Mark.
I just wanted to add a few words. It's just a real honor hosting Reto. Reto was one of the first Janelians I ever met when I was a postdoc in Texas. So it's a real honor, and having worked with Reto, I think it's going to be an excellent talk. I'm really excited to see how, yet again, I think you've managed to turn a mathematical concept into really elegant optical application. So without further ado, Reto, take it away.
Thank you, Brian. Mark. The honor is mine to present to you both Janelia and EMBL, both institutions I value highly. And yes, my talk today is about improving the spatial-temporal resolution. I could have said light sheet microscopy, but as you see in the title, it's oblique plane microscopy, which is a subfield of light sheet that just caught our attention in the last few years. And as such, with such instruments, you can, for example, in a light sheet format, acquire images of cells with finer detail than what you usually can do in a light sheet, like here, this cardiomyocyte. So I will talk about our spin on oblique plane microscopy by combining it with structural illumination, which essentially can double the resolution in light sheet. And then in the second part, I will talk about, can you actually hear me?
Perfect.
Yes, perfect.
Okay, good. Yeah. In the second part, I will talk about multi-angle projection imaging approach that we discovered based on some mathematical transforms that we essentially rediscovered and implemented in an optical computation format. And that allows, I call it here, volumetric interrogation, because the projection is not necessarily 3D imaging. It maps the 3D scene onto 2D, but I will also show extensions, how you can gather three-dimensional information from a small set of such projections. And finally, the two concepts can also be synergistically combined. So the oblique plane microscope with structural illumination can also be combined with the projection method. So it has been a long dream to combine light sheet and structural illumination, because it would essentially combine two emerging fields of optical microscopy, the super resolution field and the light sheet field together. And as I will line out, this would have obvious advantages. So what are the differences between light sheet and, say, structural illumination? Light sheet microscopy, we should all know, so I keep it brief. You illuminate your sample with something that resembles a sheet of light, so that you only illuminate the focal plane that is in view of, say, this lens up here. So you avoid exciting everything above and below your focal plane, and as such, you achieve a physical optical sectioning, not computational, but really intrinsic optical sectioning, and as such, you can lower phototoxicity and also achieve high temporal resolution. So here, I think this is a video from Philip Keller's group that shows embryogenesis. That is one field where light sheet microscopy has become really popular because you can image for days without signs of phototoxicity. These embryos will actually hatch and be fully viable, and this wonderful dance of these cells can be visualized with great spatial with great signal to noise ratio, and this would not be possible if, say, a confocal. The drawback of a light sheet is that it has a rather modest spatial resolution. Its detection is basically wide field limited, and the choice of lenses is also limited. You cannot put a turf lens or really high NA lens up there because you kind of need to share the solid angle also with the excitation objective. Structural illumination, on the other hand, is very popular is a very popular super resolution technique because it's quite biocompatible. It has good spatio-temporal resolution, so it maybe not has the highest spatial resolution, but its ability to also acquire a video rate makes it capable for time-lapse imaging, also fast dynamics. And it works with any fluorophore, so you don't need specialized fluorophores or dyes or sample preparation, so that makes it also widely deployable. It works on the principle of frequency mixing. So in the Gustafsonian sense of structural illumination, you illuminate your sample with sinusoidal interference pattern, you change the phase of the pattern and also the orientation angle and acquire data under this structural illumination. And if you do it right, you can essentially double the support in reciprocal space. So this here is the wide field optical transfer function. And when applying 3D structural illumination, you can essentially in each spatial dimension double the extent over which you can gather spatial frequencies. And here, this was early work by Lothar Schermerle and Mats Gustafsson on chromatin and the nuclear envelope, and you can see the great improvement in detail that this doubling of the OTF affords. The drawback of structural illumination, at least in its 3D form, is that it illuminates the sample essentially floodlight beam. There is no physical optical sectioning, so it's the opposite of light sheet. It essentially illuminates everything at any time. And if you have, say, a densely labeled sample, the out of focus light that you excite, that can cause artifacts. And given that you have no physical optical sectioning, you will also more quickly bleach the sample. So obviously, as I hinted before, it would be great if one could combine the positives of both techniques. Then you would have a gentle and also potentially rapid super resolution technique or if you spin it the other way around, you could essentially double the resolving power of a light sheet microscope. And why haven't we done that? Well, in a way, it has been done. So in the Lattice light sheet publication by Eric Betzig, there is data included where they use structural illumination using the lattice. And here, this is essentially the samples business end in, say, a lattice light sheet with the NA 0.7 illumination and NA 1.1 detection. And here, a 5mm cover slip that is wedged in. So if you look from the other side, you can essentially generate structural illumination with lattice light sheet, but the periodicity or the pattern vector cannot be rotated, so it's fixed. So the Gustafsonian sense for isotropic resolution extension, you would need the ability to rotate the illumination pattern by 60 and 120 degrees. So this was not possible, or has not yet been done, to do so in say, a light sheet format. What you would need, or what you could potentially do, is add additional illumination objectives. For example, here oriented in 120-degree azimuthal orientations. And if you look from the bottom, these objectives could take turn illuminating the sample with, say, a structured light sheet. This is theoretically feasible, And if I'm not mistaken, such a setup from Wesley-Ligan exists at Janelia. Not sure if this work is being in the process of being published, but nevertheless, one has to say that this is quite a complex machine. You essentially would need to weld together 3 lattice light sheet illumination trains and somehow wedge in a sample, which will become quite challenging, because if you use a glass cover slip, no matter how you orient it, it will somehow refract either the excitation light or the detection light. So it would also need, or it obviously also needs very creative sample mounting. As such, I would say if you have the means of, say, a Janelia research campus, you can maybe do it. But it would, for example, for me, quite challenging to build such a machine on a reasonable grant budget. And I would also say that sample mounting could be quite prohibitive. So let's say it's rather not practical to do it that way. So recently, we have become interested in the so-called oblique plane microscope, which essentially does the opposite. Instead of more objectives, it uses less for light sheet, or at least at the sample end. What is done in an oblique plane microscope is you use a high NA lens. We use a NA 1.35, a silicon oil lens, and you launch an oblique light sheet out of that objective. That was invented by Chris Dunsby in 2008, remained a bit dormant, and got rediscovered or re-implemented by the group of Elizabeth Hillman in 2015 by implementing a much faster scan mechanism, which was then labeled SCAPE in that particular implementation. But back to, that's just the short history of that technique. But what is going on here is that, yes, you excite, you leverage the high angular aperture of this objective to, at the very marginal rays, launch the light sheet, and then the fluorescence you detect would obviously not lie within a focal plane of this objective. So if you were to put the camera here, not much of the light sheet would be sharp. So what you actually then do is you form a remote image of the 3D scenery over here, which can be done by, at least to a theory formulated by the group of Tony Wilson, that if you match these objectives, right, you can create a diffraction limited, distortion free 3D replica of that scenery here, and as such also of any point emitting from the light sheet. So you map them over here, and then a tertiary objective can pick them up and form a sharp image on the camera. This, as I said, it has been around. It hasn't been on my radar, because one of the big disadvantages is that you use a high NA objective, but at the end you rather achieve modest spatial resolution. So typically down to the range of micron resolution, simply because you lose a lot of light. That lowers your sensitivity, but also your resolving power by essentially picking up the image at these tilted angle. This has changed a bit by Mr. Snowdy, a specialized solid immersion lens, the brainchild of Andy York and Alfred Millett-Sicking at the Calico Institute, which, again, for brevity, I will not go too much over it, but it sits here at this interface where the remote image is formed and then picked up with the tilted tertiary. This lens essentially has a theoretically very large solid angle that it can collect, and as such limits the light losses and makes the OPM concept more light efficient, but also higher in its resolving power. And so that caught our interest, and we started working on this concept. And we published this recently in eLife. And so the neat thing about the OPM is here, it can do fast imaging in the range of 13 Hertz volumetric rate of here this calcium dynamics in the neuron. And it also has decent spatial resolution on the range raw resolution of about 300 nanometer laterally. So comparable to what the lattice light sheet can achieve in its raw format. So why am I telling you this? So yes, I explained why we got interested in this, but what has that to do with the quest of combining structural illumination with light sheet microscopy? Well, what we saw was you can launch one light sheet out of the primary, but you can, in principle, also launch two out of it that diverge in an angle in that oblique plane. To better explain this, here I show schematically the light sheet coming out at an oblique angle of the objective. It diverges, but the usable beam waist is somewhere here. But in that plane, these two light sheets can essentially propagate away from each other. The angular range is limited. You cannot create arbitrary large angles, but it still corresponds to, this 64-degree opening angle between the two still corresponds to about NA 0.8. So you can virtually create interference pattern of these light sheets that are finer than what you could do with the excitation objective in the lattice light sheet. So what this does, if these two light sheets are coherent, this here is a simulation, it creates a structured light sheet. So if you were to look from the side, it would have the normal beam waist of a light sheet, but if you go along the lateral direction, it has a sinusoidal modulation. And the fringe pattern is essentially governed by the wavelengths and also by that opening angle. Sure, if we could make that opening angle larger, the pattern would be finer, but nevertheless, this allows you to get fringe spacings down to about 300 nanometer, which is not bad. It's, for SIM standards, maybe moderate, but for light sheet standards, if you also compare it to the lattice light sheet, it's quite respectable. We will see in a bit what that can buy us. But for the time being, let's just take from this busy slide that instead of one light sheet, we can also launch two, which are coherent to each other, which then in effect produce this structured light sheet. Now, how would you acquire data with this? So first, this is here shown the pupil. There is a line segment here. It's off-center to the pupil, and due to that off-centering, it creates a light sheet that is tilted. That's how it would look in an OPM. And in the structured light sheet case, you would place two mutually coherent segments out here. They would have the same offset. Well, this you can relabel this as kx and ky for the spatial frequency coordinates. So they have the same offset to the center in ky, but have an additional offset in kx. So they will both lie in the same tilted plane, but kind of are angled to each other and create that fringe spacing. By the way, here you can also see why that relative opening angle is limited, because you're already here somewhere to the periphery of the pupil. If you were in the center, you could shift them a bit further out. And so this is if you want so, a price to pay for this scheme. So if you have an NA 1.3 objective, you could maybe, if you were to place them in a normal SIM scheme to the very edge, you could maybe have a relative opening angle of 120 degrees. So you could probably create much finer patterns. But nevertheless, as I detailed before, for a light sheet format, it's still buys you quite something. Okay, and so to acquire the data, we then scan the light sheet. This has been pioneered by the group of Elizabeth Hillman, how to do that quickly, and also by others. Manish Kumar also published very neat mechanisms how you, without essentially moving any heavy objects like the sample or the objective, you can very rapidly scan the light sheet. And as such, you can then acquire volume under structural illumination. Notice it's not square, it's essentially a rectangular volume. It's essentially a parallelepiped. That's just how the data comes out, similar like in the lattice light sheet. So we would be able to acquire one data set under structural illumination. That would still not bias the isotropy. We essentially would extend the resolution in the direction of the pattern vector, but not in the other directions. So what the OPM now enables us is, though, that we can rotate that illumination pattern. Nothing prevents us from placing these illumination intensity components in different orientations in the pupil, and as such create oblique light sheets that are azimutally rotated. And so in principle, we can acquire volumes under different orientations of the pattern and then fuse that data together where they have mutual overlap and perform SIM reconstruction in the Gustafsonian sense. And so as such, this problem I outlined before, how you can, in a light sheet format, rotate the illumination pattern or the light sheet, can actually quite elegantly be solved in the oblique plane microscope case, whereas in a classical light sheet, it would be quite a headache. Oops. There is a small detail that I have neglected, but we have to address it. So here on the left, we see the two light sheets coming out of the objective, and to acquire the SIM data under different orientations, they would azimutally rotate. That is in principle not a hard technical challenge. You could think of a turf microscope with rotating illumination. There you essentially do something similar. You spin the illumination components in the pupil. However, to form a sharp image on the camera, as these light sheets rotate, their image would rotate too. And as such, this whole tertiary arm would need to co-rotate to form a sharp image of each angular orientation of the light, structured light sheet. And that would be, let's say, quite a headache, maybe on a similar scale as fusing together three illumination objectives. So it will not really be a practical microscope, I would say, and also not a really fast one. Alternatively, you could argue you could rotate the cover slip. So you could keep everything on the microscope side stationary and rotate the sample. But I would also argue this would be rather painful. By the way, if you have any questions, feel free to, interrupt. I think we have enough time. Okay, so instead of rotating the tertiary imaging path or rotating the sample, what we came up with is optical elements that can actually perform the rotation of an image. And if you don't know that, then this is actually pretty neat. This here is a so-called Abbe-Koenig prism or Dovetail prism that if you rotate it by, say, an angle alpha along its axis, it rotates the image by double that rotation angle. As you may also notice, it introduces a bias rotation. So if that Sorlapse image comes out, it's already flipped. And as you start to rotate it, it doubles that angle. The reason why the bias is there, you can imagine it that it's essentially three reflections, and an odd number of reflections causes the flipping of an image. So in principle, we could buy from Sorlapse such a prism, put it somewhere in the microscope. I will go over where one could actually place that, and mechanically rotate it. This would still be not very satisfying because mechanical rotation is still time consuming, and the block of glass is also annoying. It would cause some dispersion. It would also cause some aberrations because the light rays in the microscope don't, they have a certain divergence angle, and as they would go through that block of glass, they would probably pick up at least some spherical aberrations. So principle, doable, practically, we would still not be happy with it. However, there are alternative image rotators. One is the so-called K-mirror assembly, which are three mirrors, and I think it's called K because the reflections kind of look like a letter K if you look at this mirror and these two rays. And if you rotate this whole assembly of these three mirrors along this axis by, say, an angle gamma, then the image would rotate by 2 gamma. So the light will come in here from the left, bounce off these three mirrors, and if you rotate that whole thing, you would rotate the image. We still didn't want mechanical rotation, so a naive idea that I proposed in the lab early on is, well, let's build something like a K mirror, have this roof mirror, essentially a pair or maybe more of them stationary at different angles that we would like, correspond to the rotations angle we would like, and essentially put in 2 Galvo mirrors that then could switch passes between those, and that should do the same thing, right? It kind of looks similar like this thing here, with the caveat that the rotation angle of this guy is a little bit different than here, so it's not exactly the same. Once we did the math, you can compute the rotation matrices for such a mirror assembly. We realized that actually this angle caused quite this angular offset between gamma and alpha here caused quite a reduction in image rotation. So with practical Galvo mirrors, not much would happen. The image would rotate maybe by 10 degrees or less. So we were stuck again. So if you could rotate the light sheet by plus minus 10 degrees, you would not gain that much for a sim. So we experimented, I think, in the winter season 2020 to 2021. So in the midst of the pandemic, we had a lot of fun thinking of alternative geometries of rotating mirrors and stationary mirrors to create more rotation angle. And essentially what Bouchard and Binging in our lab came up with is this geometry, which is surprising why that works that way. But if you were, say, in the horizontal plane here spanned by X and Z, you would essentially incline these 2 Galvo mirrors and then have the stationary mirrors aligned around here. And depending how that inclination in the horizontal plane is of these 2 Galvo mirrors, it can actually amplify the image rotation angle. So here the image rotation angle is plotted, and here the mechanical scan angle of the mirrors, which is typically plus minus 10 degrees. And if the lower, the more these mirrors, the lower this opening angle of the mirrors is, the larger the image rotation becomes. And actually for around 30 degrees, you can engineer it that it exactly becomes plus and minus 60 degrees of rotation angle. Don't ask me how we figured that out. It was a bit of experimental work and then back checking the math. There was no intuition saying that this would work that way. Besides analytical solution, Bouchard also plugged it into CMAX, and maybe, given that this looks a bit dry, maybe here, if you look at this, maybe it makes a bit more sense. These 2 are the Galvo mirrors, and the red are kind of the roof mirrors that are stationary. So you have one here, one a bit below, and one a bit higher. And with the Galvos, you select to which one you go. And we also added here 2 additional mirrors to make this kind of collinear or propagating in and out along the same axis. They're not necessarily needed. You could also come in from below, bounce off, and go back out. So essentially take a U-turn with your light. But CMAX also confirmed, you don't have to do any math. It just pops out what goes in and what comes out. It also confirms that we get the desired rotation of the image, which here Bouchard just did some point cloud that then obviously rotates. This is how the thing looks in practice. It's a rather compact unit. We needed it to be compact because it has to fit into the optical train of the microscope. We didn't want to add much more optics. So as I said, we additionally wanted that it stays collinear. So the light coming in goes out in the same axis, or vice versa if it comes in from the other side. So there are 2 additional folding mirrors, but up here is essentially the 2 Galvo mirrors in that horizontal plane inclined by 30 degrees. And then here are 3 stationary mirrors, and the Galvos select over which mirror you bounce off. You could add more mirrors, or you could add less. You can also make a rotator that just goes over plus or minus 45 degrees, which could be interesting. as well, but we wanted a pure Gustafsonian rotation angles of plus or minus 60 degrees. Does that make any sense? I know it may look a bit confusing, but the real business happens with the 3 reflections of this mirror to any of these 3 static ones, and then essentially descanned by that second Galvo mirror.
So
All
Reto, each of the structural illumination patterns are then also at an oblique angle, though, right? They're not quite in plane, or do I misunderstand that?
So you can imagine the image rotator doesn't care what comes in. It could be this peace sign or well, whatever you call it, or it could be the pupil, which has, you know, not everything, but just at some rotation, a dot or a line. And so the image rotator just rotates whatever comes in. So if this comes in and you rotate by 60 degrees, then it creates the rotated version in the pupil. And actually rotation goes between Fourier space and real space. So even if the image rotator is in image space, it also rotates the same way in Fourier space or in pupil space. Does that help? So the image rotator doesn't care if the line is in the center or outside, it just rotates it.
Okay, yeah, I think that does make sense.
Yeah, but maybe in an abstraction, that thing doesn't know what it's going to be used. So there could be a Mickey Mouse distribution coming in, and it would put out the rotated version of the Mickey Mouse.
Right. I'm just trying to think ahead now to the reconstruction part, but I think maybe you'll get to that later.
That's a very good point. We will get to that too. So let's assume we have an image rotator, and as a small detail, it works both ways. It has the symmetry that you could come in from here with excitation light, and it rotates the distribution of the excitation light. And you can also come the other way with fluorescence light, and it rotates the same way the fluorescence light, both of which are necessary, as we will see in a bit, because we want to rotate the excitation, and we essentially want to derotate the fluorescence. So the symmetry is kind of welcome that it works both directions. I see. Yes, hopefully it will become clear on the next slides, else we have to iterate a little bit over it. So here we're looking straight down, say along this blue vector, straight at this mirror. And on the other side is a little barcode from a Sorlabs card or something. And here we flip between the 3 mirrors. Don't mind the translation, that's just me not holding the cell phone steady. But look, if just focus on the barcode in the middle, that's essentially where the business or the useful field of view is of the rotator, where it essentially rotates the image of that stripe pattern through the device. Okay, and this rotation is solely done by rotating these 2 Galvo mirrors, which you can do relatively fast. So these are 2 Cambridge mirrors. Their size governs a bit the field of view. So you don't want the smallest, you would want the smallest mirror you could get, but then your field of view would be limited. But here, these are like 50 millimeter wide Y mirrors of the Cambridge ones. And you may also note that they have to scan between plus and minus 10 degree mechanical angle. So that's their full angular range. So it's not a small step response, it's a big angle response, but you can still flip them with about 2 milliseconds between those states. All right, so I reckon this is pretty abstract. Maybe it becomes a bit more clear. So here is the optical path of the OPM. The excitation comes in and creates the oblique light sheet, or if it were a structured light sheet, there was just the second one behind, you wouldn't see it in this view. And the fluorescence light essentially covers the full angular range of the objective. So in pupil space, it will be the full circle, and it is mapped back into the secondary objective and then picked up by the tertiary. So in principle, the image rotator could be placed in an image space, but also in a frequency space. So in principle, it could be placed, if there wasn't a scanning galvo here, you could place it here. But this space is already occupied by the scanning galvo that scans the light sheet and descans the fluorescence. So we place the image rotator here. Could also be placed in this image space, but we chose this space here. And the neat thing is the rotator is so compact that this is a scan lens, this is a tube lens. It fits in that space. The optical path length is about 160 millimeters, and we designed the rotator to squeeze in. So no change in the optics is needed, or no relay optics. You don't need to build an additional telescope to fit that thing. So in the operation, and I apologize, I do not have a fancy animation that shows it, but essentially the excitation light comes in, it gets rotated, and as such, the azimuthal angle of the light sheet changes, and the fluorescence light returning gets derotated. So it has then again the same orientation as before and is mapped on the camera. So in a sense, you can imagine it a similar operation as the galvo. The galvo, at least in the Scape or in the SOPI by Manish Kumar, is the ingenious invention for OPM or Scape or SOPI as they're called, to essentially scan the excitation light and descan the fluorescence. So even though the light sheet and the fluorescence detection kind of shift, they're being descanned, and so the camera or not anything else doesn't actually realize that there was a shift as it's being undone. So scan and descan like a confocal. And you can think of the rotator doing the same thing. It rotates in the forward direction and derotates in the backwards direction. So the camera wouldn't even notice that it has been rotated. So in an abstract way, you could think of scanning, descanning with the galvo and rotating and derotating with the image rotator. And both of these methods allow you to keep everything else down here steady, because the light, say this path doesn't change up to here, then it gets scanned, or the path doesn't change up to here, and only then it gets rotated, and on the way back, it is undone. I have to think of a way of animating this. Maybe it will make things clearer. Let's just assume this works. And we essentially can now perform the rotation of the light sheet, and the fluorescence comes back and is always sharp on the camera. Then we can essentially acquire the data necessary for a SIM reconstruction. And the first thing we done was, I think those are 50 nanometer fluorescent beads, and compare them between OPM acquisition and OPM SIM. And if we zoom in, here, these are typical point spread functions. Let's take one of the weaker ones, because those are maybe clusters. And you can see there is, at least in the one direction, you get around 300 nanometer resolution, and you get a little bit of anisotropy in the other direction, about 340. This is expected because as you tilt this tertiary objective in one direction, you get a bit of hit. You clip some light. If you imagine here, light coming out at 60 degrees, it just sails past the snout here. And so in that direction, you will inevitably have a bit worse resolution. What I didn't mention, this drawing is actually not fully accurate. The tilt angle was actually 45 degrees. Here, this is more 30. It was deliberately chosen that way to enable finer SIM pattern. But due to that higher tilt, you get this pronounced anisotropy in the resolution. Importantly, too, is that the SIM pattern vector is actually along this direction. So the SIM resolution improvement goes along, if you want so, the good direction. So it would, if applied in a single direction, it would even further or make this anisotropy more extreme. But since we fuse data from three orientation, we overcome both the system anisotropy of the OPM and also the anisotropy introduced by the SIM process. And so here, this is the same beat, around 150 nanometer resolution, fairly round. And so we can state that essentially the doubling, if you want so, happens in the best, we can take as the baseline value the best resolution value of the OPM, which then essentially got doubled. Here, this is analysis of or kind of full width half maximum measurements over a field of beats over around 50 by 50 micron field of view, which for a sim system is around what you can get typically. For a light sheet system that sounds small, but for single cell work this is still a decent field of view. And you can see that the resolution values hover around 140 to 150 nanometer laterally, and in the CU resolution around 500 nanometer. As noted or not noted, this sim pattern is purely one-dimensional, so it increases the resolution primarily in the lateral dimension. We haven't implemented any mechanism to improve the Z resolution. You can argue, these resolution values are, if you compare them to a 3D sim, a Gustafsonian sim, they're lesser. You can argue they're maybe a square root of 2 worse than what you could do with a 3D sim, which probably if that objective could get around 100 nanometer lateral resolution and maybe 300-ish Z. However, they still compare favorably to other forms or other popular forms of structural illumination, like the SORA super resolution spinning disk or the instant sim instrument from Visitech. Here the PSFs are shown, or here is the lattice light sheet with one-dimensional structural illumination. So you haven't indicated the values. We put them on bioarchive. I should add them, but the resolution here of these kind of photon reassignment techniques is similar. So you could argue, okay, why not just using the SORA or the ISIM, which was shown later. This is still in a light sheet format, so we hope to get the benefits such as low bleaching and fast acquisition rate of light sheet in contrast to the spinning disk or instant sim. All right. Maybe let's phrase it differently. The resolution is for super resolution is okay. It's not extraordinary high, but if you, on the other hand, compare it to light sheet world, you're actually pretty happy if you get there. You usually in the light sheet world, 300 nanometer is around where you're stuck with the detection optics, and essentially doubling that for light sheet standards is pretty good. Okay, here is some cell imaging. This is MIG60 in mitochondria in U2S cells, and this should play. Here, this is the OPSIM reconstruction. There you can see there are a lot of scattered, maybe dye molecules, so these give these dots, and here we switch back to the OPM view. And the MIG60 should form small clusters in the mitochondria. They may be smaller than what we can resolve, but nevertheless you can appreciate in the OPSIM the increased level of resolution. And if we run Fourier decorrelation analysis on this cellular data, we can also see that we get about 150 nanometer resolution in a cellular context. So the bead resolution, you can argue that measurement on beads is often a bit optimistic, and it maybe is, it showed around maybe a bit lower than 150, but even in a cell context, at least single cell context, we also can get this, if you want, so doubled resolution capability. The Dylan Burnett lab got in touch with us, and they said they will have a biological structure that can serve as kind of a nano ruler, which are these myosin motor complexes. So in green are the myosin 2A motors, and in red the rod domains, and they form these assemblies that are 150 nanometers spaced of two motor proteins flanking a rod. And this, at least the Burnett lab, they have seen it with their ISIM. They could nicely resolve it. I think they're also at Geneli and used the original Gustafsson 3D sim to see these structures. And they send us a fixed sample, and we could also resolve them with the OPSIM. So this is kind of a nice biological ruler, at least for sim techniques, to show that you know, also in the biological context, get to that resolution level. I'm here, this is a U2OS cell labeled with an outer membrane protein with GFP. And here, this is the oblique plane microscopy, or if you want, a standard light sheet image, and here of the OPSIM. And if you zoom in, you can see that the hollowness of these mitochondria is resolved, whereas here in the normal light sheet mode, it isn't. So again, you could argue, okay, this is nice, but you could just use the ISIM or the SORA and get the same without all this optical wizardry, and that is correct. But what you wouldn't get is the benefit of doing that in a light sheet format. So here we imaged AP2 adapter proteins that go onto clathrin-coated vesicles, and imaged 50 volumes, both with the SORA and the OPSIM, and then compared the intensity decay of, say, the 30 brightest spots. And you can see that with the spinning disk, you much more readily bleach. And actually also the signal to noise and the signal to background of the data to start with was not by an order of magnitude, by maybe a factor of 2 lower to start with to even get there. So if you would have normalized it to the same signal to noise, it may have been even more extreme. So nevertheless, this is some hint that the light sheet format, so exciting only what you're looking at any plane, pulls through to some degree also in the light sheet imaging, sorry, in the OPSIM. The other advantage compared to the SORA is that you can now do very rapid imaging. So this is structural illumination, volumetric imaging at 1 Hertz volume rate, which would be hard just from the laser powers, say, with the SORA, and it would be hard with a Gustafsonian 3D sim, because you have to acquire a lot of data, and you have to mechanically step the focal plane. So those are rate limiting steps, whereas the OPSIM has no heavy moving parts. Essentially, all that is moving are low inertia parts like Galvo mirrors to acquire these volumes. And so as such, it is not hard limited how fast it can go. This is essentially at 1 millisecond or below 1 millisecond exposure time. It was essentially limited by how fast the camera could go. And here you can see, some, I think they have been termed nanotubes that shoot out of the mitochondria. If they're really nanoscopic, can be debated, but they still show some cool dynamics. Or if they're nanoscopic, we wouldn't see it because our resolution is still above 100 nanometer. But what you can see is that the extension and retraction gets actually quite decently resolved in space and time. Another fast dynamic or other application we could foresee is tracking clathrin-coated vesicles. So here, this is a top view and a side view of the cell. They are ARP cell that we imaged. And then we show the movie here. So here you can see the vesicles and then some zoom ins of region of interest. And this was also done at roughly 1 Hertz. And you can see that some dynamics are well resolved, but then when they really take off, there is considerable motion blurring. So this indicates even 1 Hertz might not be fast enough to track those reliably. And here in this movie, this is actually the same Roy's region of interest shown here, but also cross-sectional views. So you can see that there are some dynamics where pits take off from either side of the membrane and actually move in 3D, which is something you couldn't do if you were, for example, use a turf microscopy, which is popular in studying clathrin-mediated endocytosis. There you would just get only the bottom of the cell, and if a vesicle takes off, it leaves the evanescent field, and you wouldn't know if the vesicle has disassembled or if it just has relocated out of the turf layer. And in principle, with the OPSIM, we argue you could now study this over the entire cell, and as such, you in principle wouldn't lose a vesicle leaving your depth of focus. Okay, but it also shows the need for speed, because there is still motion blurring that is notable for the really fast moving pits. And so in principle, with faster cameras, you could probably accelerate it further, but not by an order of magnitude in a foreseeable future. So with that, we come to the second part, which is the projection imaging. However, are there any immediate questions about the OPSIM part?
You got a question from Dan Flickinger about where to place the image rotator, and he says, I would have thought the image rotator would go between scan lens 2 and its tube lens, since the field of view is smaller there, D-scan space. What is the advantage of the current location?
Yes, we have considered both. I guess a practical point was that this space is very narrow in between the scan lenses, because they're relatively short focal lengths, and the scan mirror is already there. In this space, this space was already busy, and in principle, yes, it could be placed here as well. Let me. Yes, in principle, I agree with Dan. If you design it right, you could probably create a Fourier space that could probably make do with smaller mirrors. But we simply didn't want to change too much the optical train that was working.
I think Dan meant the image space between scan lens 2 and the tube lens.
Tube lens 2.
Yes, this here.
Yeah.
There is a symmetry between the two. Actually, in the we currently use the same scan lenses here and here. But yes, this tube lens is longer. That is correct. I don't think that it changes though here, because of yes, you would have more space and more freedom to place it, but you essentially want to place it somewhat symmetrically around the image plane, so that the image is actually on the static mirrors, because else one of the galvos becomes more clipping. So at first I would think, given if that's the same scan lens, that these angles are similar, even though this adds to magnification. I don't think this space would help, but it's surely a point that one can consider. I think fundamentally there is this thing called the optical entendu. So if you create a different magnified image space, you also change the angular range, so the divergence. So even though you could kind of demagnify the image on these mirrors and say, okay, that way we increase the field of view, you increase the divergence of the image forming rays, and then they get clipped on these mirrors by divergence. So I think ultimately with the optical entendu, there is a little wiggle room, but ultimately you only effectively gain field of view with larger paddles. You can also, what you are allowed to do is change that bias rotation. So you could turn this whole assembly and essentially put it lower and make these mirrors more flat. So imagine if they were lay more in the horizontal plane, then they will become less limiting, because right now, if they're in for the central mirror are 45 degree, their width gets reduced by square root of 2. We didn't do that because we have no machine shop. So to create an assembly where these would lay down in a bathtub would have been a bit hard, but it's conceivable. Anything more, Dan? I think there are definitely things one can consider, and what I actually would love is even dispense with these scan lenses. You can also do the scanning in image space with, say, 2 galvos or a so-called scandreas. So it would be really fun to build a rotator and scanner that essentially without the scan lens, it's just between, if you want, so 2 tube lenses would do everything, because then you could gain, you wouldn't incur the light losses of these lenses.
We actually have another question from Hammer. If you want to unmic yourself or unmute yourself.
Yeah, hi, I'm Amr from Robert's Lab. I just had a very quick question. You may have mentioned this and I missed it, but in case I didn't. The 10-degree angle that you mentioned, the galvos have to do 10 degrees. Is that just to fit the field of view on the stationary mirrors?
No, to enable the rotation angle. You could come up with a solution that also would run with, so here, this 2 alpha is the optical angle. So alpha is the mechanical angle of the mirror, and then the optical angle is twice as much. And depending how you choose the geometry, so this, if you want, so opening angle of the galvos, these lines show the resulting image rotation. So I would say you would need at least practically maybe ±5 degree mechanical. But if you reduce this to 10 degree, it squeezes everything kind of together. So let's say just the overall rotation matrix, the solution, what the rotation angle is, which I haven't shown here. It's a rather ugly matrix. It's just a function of both the scan angle and the geometry angle of the system. I have to think about it too. Yes, if you make this too small, you would need a larger distance to separate these 3 mirrors. So 10 degree also works out that these fit. So sure, you could have less and just make the whole thing bigger, but that would counter our demand that it fits in that space here.
Great, thank you.
Sure. Okay. I lost any sense of time, so I hope I'm not going too slowly. But the second part, well, I would say it goes quicker, but it might be equally, what shall I say, puzzling. Puzzling sounds too negative. Equally fun in terms of, making your head hurt a little bit thinking about the mass or the optics. So what this movie here shows is essentially the end product of the second idea I want to present, which is the multi-angle projection technique. So this obviously looks like a cell that is rotating. However, neither the cell rotates nor the microscope. So this, and it's neither done by computation, this is done by optics. So we come across a transformation that in an optical sense allows us to create these views. So every view, if you go again to the start, every viewing angle here is a single camera acquisition frame. And I will, in a few seconds or minutes, explain what is going on. But this is essentially what we want to do, and here is why. So in traditional fluorescence microscopy, we acquire a 3D stack. So we're not like the computed tomography guys. We don't rotate the sample and acquire projections. We essentially acquire a focal series. This is just how we do optical microscopy. And here is the seminal paper by David Agar and John Sedat in an optimistic sense. If you just move the detection objective or the sample, you get these optical slices. We know in a wide field microscope, they don't come out as nicely sectioned, but this is the idea, and also the mass underlying it is sound, if you want so. So we would acquire the 3D stack, but as humans, we're really poor at interpreting 3D data. If you just had the 3D stack, it wouldn't make that much sense. You can just look at one 2D plane at a time. So what we usually then do is plug the 3D stack into a graphics program and maybe rotate it to look at the sample from different views. So for example, here we can see that this cancer cell has some projections or some protrusions and maybe lesser on the top, but nevertheless, that's then the visualization step, or maybe do some computations from there. In terms of understanding the data or understanding the sample, this is a time-consuming step because often it has to be done offline. It can be intense in computational resources. So light sheet microscope can easily crank out gigabytes of data in acquisition run. And it's also harder and harder to interact with really tens to hundreds of terabyte data. So if we were just interested how stuff looks, could we avoid acquiring the stack and essentially project or get projective views like here done in this graphics program? Could we directly get them from the microscope? So then this would obviously be faster because it could be done in real time, and it would also create less data. And if we could come up with some algorithms, we could maybe also infer three-dimensional information from it. And as the thing goes really fast, we could also follow dynamics that are too fast for classical 3D acquisition. Think of the OPSIM I showed before. If we really went to the lowest exposure time, some of the clustering dynamics were still too fast. So maybe if we could get an order of magnitude quicker, that could help. And so what we found that could provide us with this projective views under different angles is the shear warp transform. We essentially rediscovered it in a way. We first thought that we accidentally stumbled on it, but we then found some papers. I hope I cited below. No, my apologies. On this slide, I do not, but it was essentially the same guy who invented the what is it called? The light field microscope. He also has a paper on computer graphics using this here, the so-called shear warp transform, to compute different views. So assume here this image of a head, maybe from a CT tomogram, is the image we want to look at, or at least project from different angles. So we could obviously rotate that head and compute a projection that would give us this profile here. But bizarrely, you can also shear that image and then project it. And then you get also profile that looks eerily similar. It's just a little stretched out. But if you compensate the stretch and overlay them, and this was just done in MATLAB, you get the same profile, which is kind of weird.
I just want to remind you that your time, you're getting towards the time limit, which is fine, of course, but we want to have a little bit of meet the speaker time too. So
yes,
just
all right.
reminder.
I can speed it up. Okay, there was a little bit too much discussion. So let's skip the mass, but what we can do is essentially we can project by rapidly scanning the focal plane, and then we will get a projection along that scan direction. And what the shear warp transform allows us is we could also shift or translate laterally as we sweep the focal plane. And if we sum this, then we get a projection under a different angle. If we were to acquire a stack, this wouldn't help that much. But you can conceptually do these things also during the integration of one camera frame. Let's just skip all this stuff. This is shown here. So here, this was just purely done with analog electronics. So a signal generator scanned this cell quicker and quicker, and essentially just sweep through its focal plane. And then you get, in the time average, when you approach the frequency of the camera, one projective view. And let's play the movie again. But now we sweep the focal plane, and we also, with some galvo mirrors, translate at the same rate the image on the camera. And as we increase the frequency of these two sweeps, when it approaches the limit of the camera, you can see a different view of the cell appears. And if we change that translation factor, that shear factor, the image seemingly seems to rotate. And this is schematically shown here. You can all do that also mathematically. If you have a stack, you can just sum it along the z direction, or you can take the stack and just displace the individual image frames and sum it, and you get, thanks to the shear ward transform, a scaled and rotated view. That is the same as if you were to rotate the stack, but we don't want to do that because we want to optically do that. Sure, you can do it mathematically in MATLAB or Python, but we also want to do it optically and during one camera frame. And given that I'm short on time, I'll just skip on a few things. Here, this is in the vasculature of a zebrafish. So each of these views is a single camera exposure. You can obviously do quicker stuff. So here, this is the blood flow through a heart, where this was done. I think the raw acquisition rate was even higher, but we changed the view angle. So the effective rate was 66 Hertz, and you can get two views of the blood flow through the heart. And here, this was at roughly 120 Hertz acquisition rate. This is a cultured neuron labeled as GCAM firing. And we still changed also slowly the viewing angle. Given that I'm running out of time, you can also do kind of virtual reality viewing by quickly computing two different views of the sample. And if you had 3D glasses, this cell will pop out. And you can also infer 3D information. Oops. Like here, these are particles, gems imaged under two different view angles. And by the displacement of the dots, we can kind of compute their 3D location in 3D space. Let me skip over this. These are two collaborators, Jonathan Taylor and Daniel Olesker, that have developed an algorithm for different purposes, for their own imaging needs, that can infer from, say, two projections or more projections 3D information. And it can also be applied, even though it was developed for other means, it can also be applied to the projective views. So from 6 or 10 views here, these are mitochondria. On the cover slip shown in Z, you can infer 3D information, or here it's a bit a larger cell. It's not yet as crisp as if you acquire a true stack, but you can kind of see that 3D information can be inferred. Nevertheless, I got to move on. And the last thing I wanted to show, we can also do with OPSIM this projection. So here are the 3 differently oriented volumes under different structural nation. And we can essentially use the projection method to map them into, say, an XY plane. So we can sweep the structural light sheet through the sample and use the projection trick to essentially squash that whole volume along the z-axis into a 2D plane. And if we carefully adjust it, that it's really projecting along the z-axis, which, if you want, so is the optical axis of the primary, all these 3 planes are, or all these projection from the 3 different orientation of the OPSIM, are in the same plane, and as such, they can be fused together. So you can also, the 3 views that we registered normally, you can squash them and register them in 2D. So you can essentially perform the volumetric OPSIM imaging in a 2D format. It's important that they're precisely projected. If their angle would differ, then stuff that is higher up couldn't be registered because it would be kind of angled away or in different location for the different views. So this is a technical challenge. Nevertheless, for the sim fans, I wanted to show this quickly. This is a 3D cell under OPSIM illumination, so volumetric illumination, but projected into a 2D plane for the 3 orientation. And you can see the sim pattern moves, it modulates the sample, and you can also see that it rotates because the image rotator rotates the image. So it seemingly rotates the image. It also squishes or stretches it a little. But if you undo the rotation angle and the scaling factor, you can see here the 3 views. And if you have enough resolution on your screen, you can actually see that the pattern runs now in different orientations, and you can reconstruct it. And so here, this is the sim reconstruction from the projected views. And here, this is a 3D OPSIM reconstruction color-coded. So you can see this is really 3D. So these mitochondria wrap around the nucleus, whereas here you can reconstruct it from projective views. So you really map the 3D space into 2D. And if you look here, you can see the similar structures. However, here they come from different C-planes. And here, this is a volumetric imaging. So, well, we have not yet increased it by an order of magnitude. So it's at 2 Hertz, or the fastest we went was about 4 Hertz. But this is fast enough for these structure for the mitochondria. Here you see one nice kind of nanotube shooting out. Sure, I guess the thing you have to consider here is this is really the whole volume of the cell being imaged. Yes, it's a projection, so you cannot know if this mitochondria is on top and this one is on the bottom, but nevertheless you see them. And we think with this projective OPSIM, we can effectively crank up the speed much further than what we could do with the OPSIM. All right, this was plenty. So I would like to acknowledge everyone in our lab and all fantastic collaborators and the funding sources. And to you all for sticking with me for the whole talk. This is the end.
Thank you, Reto. So we're going to have switch the meet the speaker session. We just want to hang around and chat with Reto. Feel free to do so. Otherwise, thank you for attending the talk.
I'm going to stop the recording.
Yeah.
Or maybe we should have one question, then we'll stop. Yeah.
Yeah, I don't know. I would like to ask actually 2 questions, Reto, if you allow.
For my side, it's totally fine. I mean,
my brain
can stick around.
My brain is still rotating around everything that you showed. Really nice work on these image rotations. I think I have a practical question and then a bit more futuristic one. So maybe on the practical side, you highlighted in the beginning quite a lot how cumbersome it would be with different objectives, right, to create the sim pattern. Mm-hmm. Have you thought actually about the other approach to OPMs, where you come with a second objective from an oblique angle? Would that allow you to in a way to also achieve the same type of rotating sim pattern? Would that help in a way?
So you would add an additional you would do OPM, but you would allow a separate excitation objective.
Mm-hmm. And then, I mean, it would again be cumbersome, but maybe not so much for the sample anymore.
I think it could probably add angular aperture. So the OPM is still a bit, even with those high NA objectives, hard strapped in what angles it can run. So I would at first, without thinking too much about it, yes, it could probably open up angular space that you cannot access. If it will make it more practical, from my side, I really like about the OPM the aspect that it essentially is like an epi-fluorescence microscope. So if you bring the sample there, it essentially is aligned. As long as all the stuff down here is not drifted, then you can essentially put the sample there like it's a normal microscope. Whereas anyone who has operated Lattice or any other high-end microscope is that once you have separate objectives, if temperature changes, they change their focal lengths and walk away from each other. And so it just makes everything a bit more cumbersome. But yeah, before I ran too much, yes, I think there are interesting ideas also adding a second objective from the top, not in a 4π sense, but just to increase what you can interrogate, both in an OPM scheme, like having an opposite side view of the sample, but also on the excitation side, what you can launch into it. So if I weren't moving to an 11th floor, I might consider it.
Okay. I mean, I think the image rotation that you showed is a lot more elegant than what I had in mind. And then the second question was a bit more futuristic in the sense that you said, of course, the SIM provides image doubling in 2 dimensions. So then can you sort of a little bit tell us in which direction this would need to be adapted to, so you can also double the resolution in the Z direction? Does it help somehow that you're actually imaging a skewed volume in a sense? Would that
It helps and it doesn't. So without going too much into the detail, we still did the reconstruction a bit simplified. If you're aware of how what different SIM algorithms exist, there is a way of doing slice by slice. So even though data is 3D, even in a 3D SIM, you can treat it slice by slice. But that essentially, we just did that because, yeah, for a lack of having someone who really wanted to dig into the algorithmic side. It would be much better to do it in kind of a multi-view fusion approach. So accept that these are different volumes that have also slightly slanted OTF and essentially fusing that data in 3D. We got away with the slice by slice because the PSFs actually surprisingly align quite well with the z-axis. That's not a given. The OPM is such a weird beast that you know it kind of skews the PSF too. But here we were just lucky. But yes, you should get a benefit from fusing. You know, you could imagine if they're slightly tilted to each other by properly fusing them in 3D, you get a little advantage. And the group at Chris Dunsby, I think they did a dual view OPM with a different mechanism. And they fused, I think they 3D fused or used the image fusion algorithm that helped. But that alone will not give you, you know, a resolution doubling.
Exactly, yeah, that's what I was wondering. Sort of what would be.
But it would be better. It would be better. So there is still something you can improve in Z by properly treating it. And you can, of course, also implement an optical lattice in the sense that you also have periodicity, not only in one dimension, but in the other. The pupil space doesn't allow you to create the same as, you know, let me go, where is their slide? You know, the additional dimension, let's call it KY, you cannot go symmetrically, you know, plus and minus to for example, create something like a hexagonal lattice. Its aspect ratio would be, you know, different because here you simply lack space. You can go further out here to create the lateral pattern, but lesser in the other direction. But still, you could gain something. My guess is that the game would still be lesser than what you could get in a 3D sim. So what we're actually excited about at the moment is photo switchable fluorophores that we essentially, I don't have a slide in here, but we essentially create positive and negative light sheets to essentially activate the layer of fluorophores and then thin that layer with a deactivation light sheet that has a 0 around its center plane. So I personally think this is attractive. Sure, it's not attractive in the sense that you then trade away the advantage of SIM being applicable to all fluorophores, but it doesn't need, you know, it's not as hard limited on this K-space coverage of a traditional linear SIM. So we're experimenting with that, if I can lean out of the window. And let's say that. Yeah.
No, sounds really cool. I mean, looking forward to what comes next. So yeah, thanks a lot.
Sure.